Now the chapter 18, that is neural control and coordination. So we are going to study the neural system, human neural system, then neuron as structural and functional unit of neural system, then central nervous system. So as you know, the functions of the organs and organ system in our body must be coordinated to maintain homeostasis. Now coordination is the process through which two or more organs interact and complement the function of one another. Okay, so in here, like, nothing is that important. This line is important. Like, in our body, the neural system and the endocrine system jointly coordinate and integrate all the activities of the organs so that they function in a synchronous fashion. Okay, the neural system provides an organized network of point to point coordination for a quick coordination. So this is quick coordination for the neural system, that is point to point. And then endocrine system provides chemical integration through the hormones. Okay. Okay, so in this chapter we are going to learn like neural system of human, mechanism of neural coordination, like transmission of nerve impulse and impulse conduction across a synapse, like across a synapse. Right, neural system. The neural system of all animals is composed of highly specialized cells called neurons, which can detect, receive, and transmit different kinds of stimuli. The neural organization is very simple in lower invertebrates. For example, in Hydra, it is composed of a network of neurons. The neural system is better organized in insects, where a brain is present along with the number of ganglia and neural tissues. The vertebrates have a more developed neural system. Now the human neural system. The human neural system is divided into two parts: the CNS and PNS. So CNS, that is central nervous neural system, and PNS, that is peripheral neural system. The CNS includes both the brain and the spinal cord and is the site of information processing and control. The PNS comprises of all the nerves of the body associated with the CNS, that is brain and spinal cord. So all the nerves that is associated with the CNS, that is brain and spinal cord, these are the PNS. And the nerve fibers of the PNS are of two types: the apparent nerve fibers and efferent fibers. The apparent nerve fibers transmit impulse from tissues organs to the CNS, and the efferent fibers transmit regulatory impulses from the CNS to the concerned peripheral tissues and organs. So apparent takes from tissues to CNS, and efferent takes from CNS to tissues, right? Tissues. The PNS is divided into two divisions. So two divisions, like somatic neural system, autonomic neural system. The somatic neural system relays impulses from the CNS to skeletal muscles, while the autonomic neural system transmit impulse from CNS to involuntary organs. So CNS to the skeletal muscle, that is somatic neural system, and CNS to involuntary organs, that is autonomic neural system. So and smooth muscles of the body. Okay, involuntary organs and smooth muscles of the body. The autonomic nervous system is further classified into parasympathetic neural system and sympathetic neural system. So visceral nervous system is the part of the peripheral nervous system that comprises the whole complex of nerves, fibers, ganglia, and plexus, by which impulses travel from the central nervous system to the viscera, and from viscera to the central nervous system. So visceral nervous system, from viscera to the central nervous system to viscera, and from viscera to central nervous system. So just remember that. So neuron has functional so neuron is structure and functional unit of neural system. Now, neuron is a microscopic structure composed of three major parts, namely cell body, dendrites, and axon. The cell body contains cytoplasm with typical cell organelles and certain granule bodies called Nizels granules. Short fibers which branch repeatedly and project out of the cell body also contain Nizels granules and are called dendrites. So these fibers transmit impulses towards the cell body. Now these are the dendrites. Axon is a long fiber, the distal end of the distal end which is branch. Each branch terminates as a bulb-like structure called synaptic knob, which possess synaptic vesicles containing chemicals called neurotransmitters. So synaptic knobs which possess synaptic vesicles, these are the chemicals called the neurotransmitters. The axons transmit nerve impulses away from the cell body to a synapse or to a neuromuscular junction. So to a synapse or to a neuromuscular junction, that is motor neuron plus muscle fiber, there is a junction. Like, based on the number of axons and dendrites, the neurons are divided into three types: multipolar with one axon and two or more dendrites, found in the cerebral cortex. So multipolar with one axon and two or more dendrites, found in the cerebral cortex. So okay, bipolar with one axon and one dendrite, found in the retina of eye. So bipolar, like you see, like two eyes and two. And then you can see the unipolar, the cell body with one axon only, found usually in the embryonic stage. So unipolar cell body with one axon found usually in the embryonic stage, right? Only one axon, no dendrites, right? So there are two types of axons, namely myelinated and non-myelinated. The myelinated nerves includes one cell which form a myelin sheath. So okay, so I give you like that. They are sitting in here. So So there are two types of axons, namely myelinated and non-myelinated. Myelinated and non-myelinated. The myelinated nerve fibers are enveloped with Schwann cells, which form a myelin sheath around the axon. The gaps between the two adjacent, like the gap between the two adjacent, the gap between the two adjacent myelin sheaths are called the nodes of Ranvier. Myelinated nerve fibers are found in spinal and cranial nerves. So myelinators are found in the are found in the are found in the are found in the spinal and cranial nerves. Unmyelinated nerve fiber is enclosed by the Schwann cell that does not form a myelin sheath around the axon and is commonly found in autonomous and the somatic neural system. So unmyelinated nerve fiber, they are present in the autonomic and neural somatic neural system. So autonomic and neural somatic system, as you can just remember that, like, so unmyelinated nerve fiber is enclosed by an Schwann cell that does not form a myelin sheath around the axon and is commonly found in autonomous and the somatic neural system. Autonomous and the somatic neural system, right? Okay. And myelinated nerve fibers, they are found in the spinal and cranial nerves, right? Spinal and cranial nerves, you can see, right? So generation and conduction of nerve impulses, so you can see this is so this is the structure of neuron. You can see the dendrites, they transmit the impulses toward the cell body. Then nasal granules, these are present. These are also present in dendrites and in the cell body, as you can see in nasal bodies. Then there is cell body that contains the nucleus, you can see. Then there are Schwann cells, and then there is the axon, and then there is a myelin sheath. Then there is a node of Ranvier, then axon terminal, and then the synaptic node, but that contains the chemicals, which is a neurotransmitter, right? So this is it. And then the generation and conduction of nerve impulses. Neurons are excitable cells because their membranes are in a polarized state. So neurons are the neurons are the excitable cells because their membranes are in a polarized state. Do you know why the membrane of a neuron is polarized? Because different type of ion channels are present on the neural membrane. So they allow the different types of movements. So they allow the different types of concentration of movements that gets exchanged. So this is why there is always a difference. So this is why they are polarized, right? So these ion channels are selectively permeable to different ions. So these ion channels are not selectively permeable to the same ions, but they are different ions. So when when the neuron is not conducting, when the neuron is not conducting any impulses, that is resting. When the neuron is not conducting any impulses, that is resting. The axonal membrane is comparatively more permeable to potassium ion, that is K+, and nearly impermeable to sodium ion. So it is more permeable to potassium ion, then it is almost impermeable to sodium ion. So similarly, the membrane is impermeable to negatively charged proteins present in the exoplasm. So impermeable to sodium ion, and the negative charge, to the negative charge, where to the where to okay so negatively charged proteins present in the exoplasm. So consequently, the exoplasm inside the axon contains high concentration of potassium and negatively charged proteins and low concentration of Na+. In contrast, the fluid outside the axon contains a low concentration of potassium and a high concentration of Na+, and thus forms a concentration gradient. So these ionic gradients across the resting membrane. So these ionic gradients across the resting membrane are maintained by the active transport of ions by the sodium potassium pump, which transports 3 Na plus outwards for 2 K plus into the cell, right? As a result, the outer membrane of the axonal membrane possesses a positive charge. So as a result, the outer surface of the axonal membrane possesses a positive charge, while its inner the outer the outer because you transport 3 Na. So of course the positive charge is more, right, than the negative charge. So as a result, the outer surface of the axonal membrane possesses a positive charge, while its inner surface becomes negatively charged, and therefore is polarized. So therefore the membrane is polarized. Now the electrical potential difference across the resting plasma membrane is called as the resting potential. Now we are going to study the mechanism of nerve impulses, so its conduction along an axon. So when a stimulus is applied at a site, that is point A, on a polarized membrane, the membrane at the site A becomes freely permeable to Na plus. Firstly, outside there is Na, and inside there is potassium ion and some negatively charged, right? Now when the stimulus is applied at A, becomes freely permeable to Na plus. So it becomes freely permeable to Na plus. So this leads to a rapid influx of Na plus, followed by reversible of polarity at the site. Okay, that is the outer surface of the membrane becomes negatively charged because Na plus gets inside into the membrane, becomes negatively charged, and the inner side becomes positively charged. Right, the polarity of the membrane at the site A is thus reversed, and hence it is depolarized. The electrical potential difference across the plasma membrane at the site A is called the action potential because it is depolarized. So depolarization means the action potential is generated, which in fact term as a nerve impulse. So action potential, it is a nerve impulse, it is like the depolarization. Okay, so depolarization, action potential, nerve impulse is the same thing. So as such, immediately ahead, the axon at B membrane has a positive charge on the outer surface and a negative charge on its inner surface. Has a positive charge on the outer surface and a negative charge on the inner surface. Okay, so at B, as a result, and then there is all the current flows to complete a circuit and all that thing. Hence the polarity at the side is reversed and action potential is generated at B. So from A to it gets converted to B, then thus the impulse, that is action potential, generated at side A arrives at side B. The sequence is repeated along the length of the axon, and consequently the impulse is conducted. The rise in the stimulus induced permeability to Na plus is extremely short lived. The rise in the stimulus induced permeability to Na plus is extremely short lived. The rise in the stimulus induced permeability to Na plus is extremely short lived. It is quickly followed by a rise in permeability to K plus. So when the Na plus gets entered into the axon, it is very short lived. It is quickly followed by the rise in the permeability to K plus. So within a fraction of a second, K plus deposits outside the membrane and restores the so K plus diffuses outside the membrane and restores the resting potential of the membrane at the site of excitation, and the fiber becomes more responsive to further stimulation. So when now after that, when okay, so first Na plus gets into the axon, and in a short immediately the K plus gets outside. But to maintain this, there is a sodium and potassium pump. Now what it does, like, to maintain it again, so it throws out 3 Na plus outside, or 2 K plus, 2 K plus inside, and that means it again maintains the polarization. So now, so yeah, that's it. Now we are going to like transmission of impulses. Now another impulse is transmitted from one neuron to another through junctions called synapses. A synapse is formed by the membranes of presynaptic neuron and a postsynaptic neuron, which may or may not be separated by a gap called the synaptic cleft. Right, synaptic cleft. There are two types of synapses, namely electrical synapses and chemical synapses. At electrical synapses, the membrane of pre and postsynaptic neurons are in a very close proximity. The electrical current can flow directly from one neuron into the other across the synapses. Transmission of an impulse across electrical synapses is very similar to impulse conducting along a single so just suppose that a single axon, and that's the electrical synapses. What it does, like, it is very rarely, synapse is very similar to impulse conduction along a single neuron. Impulse transmission across an electrical synapse is very faster than that of a chemical synapse, because there is no synaptic gap. They are very close in. So electrical synapses are very rare in our system, although. So at a chemical synapse, the membranes of presynaptic and postsynaptic neurons are separated by a fluid-filled space called synaptic cleft. Do you know how the presynaptic neuron transmits an impulse action potential across the synaptic cleft to the postsynaptic neuron? It is by the chemicals. Okay, yes, I know. Chemicals called the neurotransmitters are involved in the transmission of impulses at these synapses. The axon terminals contain vesicles filled with these neurotransmitters. When an impulse, that is action potential, arrives at the axon terminal, it stimulates the movement of the synaptic vesicles towards the membrane. So when the impulses gets in at the axon terminal, it stimulates the movement of the synaptic vesicles towards the membrane, where they fuse with the plasma membrane and release their neurotransmitters in the synaptic cleft. The released neurotransmitters bind to their specific receptors present on the postsynaptic membrane. This binding opens ion channels, allowing the entry of ions which can generate a new potential in the postsynaptic neuron. So this binding opens ion channels, allowing the entry of ions which can generate a new potential in the postsynaptic neuron. Now this can be inhibitory or excitatory. So in the next part we are going to study the central neural system. So like this is it. And you can see in the diagram there is an axon, then is an axon terminal, that is axon terminal, it has like axon terminal, then it has a synaptical vesicles, then there is a presynaptic membrane, then is synaptic cleft, then the postsynaptic membrane. So postsynaptic membrane has the receptors, okay, on the postsynaptic membrane. Now, and there are neurotransmitters get attached to the receptors and all that. So synapses is formed by the presynaptic membrane and synaptic cleft, left and postsynaptic membrane. So all this like showing an axon terminal and synapses. So this is it.